Yep. Right. Thank you for the kind introduction. Hi, everyone. So as you introduced, my name is Ki-hoon Jeong. I'm from Seoul National University in Seoul, Korea. So as you also introduced, I'm on sabbatical at Stanford in California. So it's not too early morning here in California, not in Korea time. So which is good. So I'm gonna talk about the role of immune cells, especially some myeloid cell populations and tumor the microenvironment, especially in colorectal and pancreatic cancer, and most recently cervical cancers as well. So for a brief introduction, so as you are see here, so there are the various types of cancer therapies available in the clinic. So previously, there were more attempts to develop therapies to kill the cancer cells directly, such as chemo or radiation therapy or the so-called the targeted therapy. However, these days there are more and more drugs being developed to target this tumor, the microenvironment, such as immunotherapy and anti-angiogenic therapy, and many others like this. Among these, given that the anti-angiogenic therapy is a standard of care for multiple types of cancers, today that I'll share some of the results with targeting the angiogenesis. As you are familiar with, so among many angiogenic factors, the VEGF signaling pathway, the vascular endothelial growth factor, the VEGF signaling pathway, is a key component of pathological angiogenesis in most cancers. Supporting these facts, so far more than 10 different anti-VEGF drugs have been approved by the FDA to treat various solid tumors in the clinic. Starting with metastatic colorectal cancer back in 2004 as the first indication. However, the survival benefit from this drug is quite disappointing in practice. So because tumors develop the resistance to these drugs. So there have been a couple of potential mechanisms of the resistance proposed so far. So among these, today I'm going to focus on the role of the immune cells in the resistance process. So these are the typical immune microenvironment in cancers, which is characterized by abundant infiltration of lymphoid and myeloid cell populations. And also as tumors grow, some of the immunosuppressive cells are expanding, such as FOXP3 positive regulatory T cells or so-called M2 polarized tumor-associated macrophages. And these immunosuppressive cells are the major players for the immune escape of the tumors because these cells basically suppress the anti-tumor immunity and help tumors avoid the host anti-tumor attack. And also one big problem in the field is that since the immune cell the migration and their interactions are too highly dynamic, especially when it comes to in vivo, so the immune microenvironment is actually pretty difficult to study using conventional experimental techniques that just only allows very static analysis at certain time points. So we needed new methods to monitor such dynamic behaviors of the immune cells in vivo and the real-time manner as well. So this is example. So it's showing that this is a monocyte rolling along the vessel wall and some flowing cells inside the vessels. So all of these are being played in real time. So and therefore, so I would say this system is quite suitable for the immune cell trafficking in vivo. So I'm not going to talk about too much detail of this imaging setup of this system, but let me just briefly introduce how I visualize. So for example, these videos were acquired from the beating heart, live mouse beating heart. So to do so, some years ago, we developed a in vivo imaging method, basically endoscopy imaging method to visualize and monitor the beating heart. So as you can appreciate here, this is a very thin imaging probe made of green lens, the graded refractive index lens, so that we can insert through the intercostal space, basically between the ribs. This is thin enough to be minimally invasively inserted through the chest cavity. So that way we can still make the mice alive. So the mice are just anesthetized, just sleeping during the imaging session. So we can do the live imaging, and then we can also do the longitudinal imaging, like a day-by-day imaging. using this system. To stabilize the motion fact from the beating heart per se, so we also equipped our imaging system with the micro suction system, so that way we can give the negative pressure on the surface of the beating heart. That way we can stabilize the beating heart tissues. So we can also implement some larger the tube so that way we can achieve the wide field imaging of the beating heart like this. So eventually what we were able to do was like this. So we basically obtained this large area, the imaging of the whole the vasculature of the surface of the beating heart like this. So using this system, we also were able to apply it for some disease model. For example, so this is a myocardial infarction model of the live mouse. So that way you can see that this infarct area, there are much more the immune cell, the migration, like a this particular case was the monocyte. We can also do the longitudinal trafficking of the monocyte inside the infarct and compare the remote area, the healthy area like this. So again, this is just summary of this, the imaging setup of this that we developed for the beating heart. So like I said, this is good enough for immune cell trafficking in vivo. So having these techniques available, we studied the role of the immune cells in this anti-VEGF therapy, the resistance process in a tumor, the microenvironment. So first of all, we wanted to obtain some clinical insight. So we looked for potential immunomodulator factors responsible for the resistance process. So previously, my former group analyzed rectal cancer patient samples before and after the bevacizumab treatment, which is anti-VEGF therapy. So among many factors, we found a huge increase in expression of chemokine receptor, which is CXCR4 receptor, and its ligand CXCL12, which is also known as SF1 alpha. So the next question was then, what is the role of this unregulated CXCR4 pathway in this after the anti-VEGF therapy? So given that the CXCR4 chemokine receptor is important for the leukocyte trafficking, the chemotaxis, they analyzed the various immune cell populations in the colon cancer, the microenvironment, and found that this particular chemokine receptor was mainly expressed by the monocyte and neutrophil. So we further investigated these myeloid immune cells in this colorectal cancer, the microenvironment. So in this FACS plot, this lysis-G positive population is so-called neutrophil, and this lysis-G negative and lysis-C high population is the classical monocyte, or we can also call them as lysis-C high monocyte. In addition to these two populations, we also identified this lysis-G negative and lysis low population, which we can call the non-classical monocyte or just lysis low monocyte. So we analyzed these populations that express CXCR4 chemical receptor on their surface during the course of the anti-VEGF therapy. So the 5 days after the DC101 treatment, DC101 is the anti-VEGF-R2 antibody that we used as an anti-VEGF therapy reagent in the mouse model, and C is the control IgG. So as you can appreciate here, there was a selective increase in tumor infiltration of this non-classical monocyte compared to control. So the second row is basically classical monocyte, and third row is the neutrophil populations. As time goes by, on day 12 after we treated DC101, we've observed a further increase in non-classical monocyte, and we also observed a similar kinetic response in the other types of orthotopic tumor model we use, CT26. So basically, overall, these data suggest that basically non-classical monocyte respond to the anti-VEGF therapy first, and then they infiltrate into the tumor tissues. So next, we asked how these cells, these non-classical monocytes, are recruited to the tumor tissues. So to investigate the dynamic infiltration of the monocytes, we utilize intravital, the multiphoton microscopy. So to do so, we developed a abdominal imaging window for this study, so like this. So through this metal ring-based window, so we can visualize a green color labeled monocyte inside the post-capillary venule in a normal cecum here left side and in the tumor tissue as well to the right side. So using this system, this is actually day 12 after we treated DC101, which is anti-angiogenic therapy. So again, this is a video being played in real time. So in addition to this crawling and rolling the monocyte slowly moving cell inside the vasculature, we were also able to monitor the fast moving, the flowing cells in the bloodstream thanks to the video red scanning system, which is fast enough to capture such dynamic migrations. So in animals treated with DC-101, again, anti-VEGF-R2 antibody, so we frequently observe these green monocyte freely flowing in the blood that then began to interact with the vessel wall, either rolling or crawling, and some of the crawling cells eventually extravasate out of the blood vessel. So basically, we were able to visualize the process of serial transmigration of the monocyte that infiltrate into the tumor tissues. So which is also consistent with the data that I just shared with you, the flow cytometry data showing the abundant infiltration of this non-classical monocyte into the tumor tissues. So this is the quantification result of all the monocyte, non-classical monocyte trafficking into the tumor tissues. So with the different behaviors like flowing and rolling and crawling. So that way we can measure and quantify this flux of this monocyte moving, migrating into the tumor tissues. So again, the bottom line is that the anti-VEGF-R2 therapy, in other words, anti-angiogenic treatment, basically induce abundant infiltration of these non-classical monocyte into the tumor, the microenvironments. So to make a long story short for the colorectal cancer part, so when we start anti-angiogenic therapy, although I had to skip all this detailed mechanistic part, so first, the blockade of VEGF signaling pathway in endothelial cells upregulates another chemokine ligand, which is CX3CL1, which is also known as fractalkine, and that attracts its receptor, which is basically CX3CR1 chemokine receptor that is mainly expressed by this non-classical monocyte on their surface. And those tumor infiltrating a non-classical monocyte secrete another chemokine ligand and recruit a neutrophil population to the tumor tissues. And these tumor infiltrating the monocyte and neutrophil basically secrete a bunch of immunosuppressive cytokines like IL-10 and TGF-beta that inhibit cytotoxic T cell in the tumor microenvironment, which results in the promoting tumor growth. And that is one of the main axes of the anti-VEGF therapy, the resistance process. So we were also able to develop some therapeutic strategies by blocking this each single pathway. Basically, we also developed a combinatorial blockade in addition to this anti-angiogenic therapy, so which was all kind of successful in colorectal cancer model. So basically, so data so far that I share with you is all about colorectal cancer and only when we treated anti-VEGF therapy. So, but when we analyze other types of tumors like GBM, glioblastoma, adult brain tumor, or PDAC, the pancreatic ductal adenocarcinoma, so as you can appreciate here, there are already abundant infiltration of these monocytes in these different types of tumor, the microenvironment, so even without anti-VEGF therapy, as you can see here. So speaking of PDAC, the pancreatic cancer, some years ago, so we collected tumor and meshed blood samples from several hundreds of patients with PDAC, the Korean cohort of pancreatic cancer, and then analyzed the genome and transcriptome and proteome of these patient samples in collaboration with the Professor Huang and Lee and Zhang at SNU Hospital. So integrated clustering of mRNA and protein and phosphorylation data identified 6 different pancreatic cancer PDAC, pancreatic ductal adenocarcinoma PDAC subtypes with different survival rate like this. So among these, subtype 4, the first one, the red one, showed the worst prognosis. And let me call them as a subtype 4 as a TS4 here. And the subtype 6 with a good prognosis with a green color here in survival curve. So let me call the subtype 6 as IS2 subtype. So to explore the microenvironment in vivo, so we developed orthotopic PDAC mouse model with the cancer cell derived from the patient tissue specimens of the representative TS4 subtype and IS2 subtype, respectively. And as I said, as I just briefly mentioned, since it is an orthotopic tumor model, the tumors basically reside in the abdominal cavity. So we utilize ultrasound imaging system to monitor to non-invasively monitor the tumor growth that grow inside the abdominal cavity in the pancreas, which is again is an orthotopic tumor model. So when we first compared the tumor growth in this model, we found that TS4 tumors here, this one blue, the graph here, they grew way faster than IS2 tumors. And this is the end time point analysis and tumor volume shows that the TS4 tumors grew way faster like this. So we were wondering then what the difference is basically in the tumor, the microenvironment. So we utilize flow cytometry, the facts to examine the number of the tumor infiltrating immune cells, especially the myeloid cell populations. As you can see here, the higher level of infiltration of this polymorphonuclear PMN, MDSC, or so-called mainly composed of the neutrophil populations, which is basically granulocyte, are the were observed in TS4 tumors than in IS2 tumors in red here. And so we went back to, this is from the mouse model data. So we went back to clinical sample specimen, to patient sample specimen, and we analyzed immunohistochemistry. As you appreciate here, so CD6B is one of a granulocyte marker. So as you can see here in TS4 with the poor prognosis has much more infiltration of the CD66B population, which is a granulocyte and mainly the neutrophil populations, which was also consistent with our preclinical model. This is quantification of this immunohistochemistry. And then we also further analyze the cell population of the neutrophils. Well, I'm not going to talk about too much detail about this, but to make a long story short, what we found is that although there were many different cell population of these neutrophil populations, what we found is that the CXCR2 and R4 receptor double positive cell population of the neutrophil were the most increase in these TS4 tumors with the poor prognosis. So subsequently, we performed the in vitro CFSA assay by co-culturing this neutrophil or other myeloid cell population as a control to see if they have some T cell the regulation effect. So as you can appreciate here, although some classical monocyte CM and non-classical monocyte also exert some extent of the inhibiting effect, the neutrophil was the most potent in terms of inhibiting T cell the proliferation in this assay. So overall, these data suggest that these neutrophil populations may be in charge of depletion of this T cell population and inhibited T cell activity in this tumor, the microenvironment in the pancreatic cancer. So overall, these data suggest that neutrophil could be a promising target in the pancreatic cancer. So quite recently, similar to PDAC study, so we did proteogenomic analysis of the human LACC, locally advanced cervical cancer, basically cervical cancer patients, and we also identified 6 different subtypes. And among the treatment resistant subtype of the cervical cancer, the immune cold squamous cervical cancer subtype 5 frequently exhibited aggressive local tumor growth, and even during the course of radiotherapy. So when we analyzed the human tissues, we found that the subtype 5 tumors had the highest density of CD66B positive cells, again, the granulocyte and mostly neutrophil, compared to tumors in the other subtype, suggesting the presence of neutrophil in this particular subtype. In fact, the flow cytometry analysis showed higher percentage of IL-17 receptor positive neutrophil in subtype 5 tumors than in the bone marrow and spleen and blood also. And then we also performed the chemotaxis migration assay, showing that these IL-17, interleukin-17, strongly induce the recruitment of the neutrophils indeed. So overall, these IL-17 pathway seems very important in terms of trafficking of the neutrophil and the activity of neutrophil in the regulation of the tumor, the microenvironment. And similar to the PDAC study, we also performed in vitro CFSA assay and found that neutrophil significantly inhibited the T cell activity also. So overall, the neutrophil could be really a promising target, not only in the PDAC model, the pancreatic cancer, but also in the cervical cancer model as well. So we further actually analyzed the pancreatic cancer microenvironment by performing some single-cell RNA sequencing to to further study this population of the immune cell and what kind of cells express which factors. So as you can appreciate here, to us, very interestingly, the PDL1, which is one of the most famous immune checkpoint molecules, which is the target of atezolizumab, the antibody. So basically, PDL1 was mainly expressed by the myeloid cell populations, especially in the neutrophil population like this. So we thought that PDL1 might be a good target to block the neutrophil and inhibit tumor growth as well. And at the same time, so as you are all aware of this, so since there are many clinical trials ongoing lately that combines anti-PD-L1 and anti-VEGF therapy, so we aim to target both PD-L1 and the VEGF simultaneously, which could be effective in treating PDAC as well, the pancreatic and cervical cancer as well. So, in collaboration with a structural biologist at KAIST, Professor Ho-Min Kim, so we developed a novel fusion protein, which is called ATEG-REF, since ATEG from atezolizumab. So basically, this yellow part is a single chain F3 of the atezolizumab antibody that targets anti-PD-L1 antibody that fused to the N-terminus of the VEGF GREB portion. VEGF GREB is basically the soluble decoy receptor of the VEGF receptor. So that basically sequesters all the ligand of the VEGF superfamily proteins. So this recombinant protein ATEG-REB, so we successfully generated, and then we confirmed that it nicely forms the dimer in this Kumasi staining in the non-reducing condition. And then we also confirmed that although it is a fusion proteins, basically it still has the binding capability to the binding partner of the ligand, like PDL1, and also all the different VEGF superfamily ligand protein, like VEGF and PLGF also. So basically, so this fusion protein still has the capability of the binding to the original binding partner. So this is quite this motivated us to move on for the in vivo experiment. So to do so, we basically combine this ATEG-REB. ATG is the ATEG-REB, the fusion protein that we generated, and then we combine with the gemcitabine, which is a standard of care for to treat pancreatic cancer these days in the clinic. So when we combine these two treatments, so we were able to find a synergistic effect of the ATEG-REB compared to gemcitabine alone. So the next question was then obviously what is the mode of actions? How this synergistic effect was achieved by the treatment of these two therapeutic regimens. So although I had to skip all this part in the pancreatic cancer part, but then so let me just briefly tell you about why it's so difficult to treat pancreatic cancer compared to other types of tumors is that one of the main feature of the pancreatic cancer is the tumor fibrosis. Basically, tumor tissues are very dense and very stiff. So that way, all the blood vessels inside the tumor tissues are very compressed. Then there's almost no perfusion that induce hypoxia, and that hypoxia basically induce all immunosuppression and even more tumor fibrosis. It basically comes into the vicious cycle. So measuring the tumor fibrosis is very important in terms of studying the pancreatic cancer, the microenvironments. So to do so, we wanted to evaluate the tumor fibrosis by measuring the collagen deposition through the second harmonic generation using the two-photon the microscopy that I briefly introduced. And as you can appreciate here, compared to control, the gemcitabine the single treatment only, it's just chemotherapeutic, induce a bunch of collagen deposition like this. But then when we combine this ATEG-REB, the fusion protein in this model, we were able to observe a significant reduction of these collagen depositions. Basically, majority of the tumor fibrosis were relieved. And then, so this is basically one of the main reasons how we achieve this anti-tumor effect of this two combinatorial treatment, basically. And then let me just skip this part. But basically, by running some single cell RNA sequencing, we were able to identify the population of the calf cancer-associated fibroblasts that is in charge of promoting tumor fibrosis, which was induced by gemcitabine, and then it was just really all completely gone by the treatment of Artegrep, which is the second subpopulation of the calf, and we also identified a surface marker of this, so we call CD141 positive calf was the major suspect of this, or the increase of tumor fibrosis induced by the gemcitabine treatment. And this is all the fibrosis pathway analysis result. And since time is running out, to make a long story short, so this is the analysis of the immune microenvironment in the pancreatic cancer. As you can appreciate here, by treating, so it's yellow bar is the combinatorial treatment, which is Artegrep plus gemcitabine. So as you can see here, not only the overall T cell populations, but also basically the effector cytokine secreting T cells are much more in the Artegrep and gemcitabine treatment, which overall suggests that by the relieve the vessel compression by relieving the tumor fibrosis, there are much more blood perfusion, and the T cell can even more easily infiltrate into the tumor tissues. That way, we were able to see observe the synergistic effect by combining these two different regimens. Okay, so this is ongoing result that just basically suggests that we are identifying some novel population of the neutrophil that is in charge of skewing the tumor microenvironment toward the immunostimulatory the thing one. Okay, so time is running out. So this is summary of the pancreatic and cervical cancer. So with that, I'd like to acknowledge all the people in my lab, especially my very talented students and now all postdocs and professor also, Dr. Zhong and Dr. Kim. Ju and Ducky did a lot in pancreatic cancer work and cervical cancer. And all my collaborator in the hospital, so they provide all the different types of cancer patient specimens. So again, I appreciate guys. Thank you for all the patients and our collaborators. And yeah, thank you for your attention. Yeah, thank you.
Thank you, Professor Jung. So let's move on to Q&A session. So there's one question. Have you tracked T cell infiltration? So could you
Uh,
see the question? Yeah.
well, I cannot see for some reason, but let me go back to the T cell data. I guess this one, right? So T cell infiltration in the pancreatic cancer. So yeah, I just went through quite fast. So this blue bar is a gemcitabine treatment only group. The grayish is the control, and the yellow is the combination, the group basically. So what I just briefly said, and let me explain a little more, is that so basically, overall CD4 and CD8 T cells were much more in the tumor tissues of this combinatorial treatment group. And then not only the number of the T cell, but then the interferon gamma secretion and perforin secretion was much more in this combinatorial group as well. And also, very interestingly, PD-1 positive, which is called exhausted T cells, are much lower in this the tumor microenvironment. So I hope this answers the questions.
Yeah. Thank you for the answer. And we'll now transition to our second session. Professor Kim, could you share your slides?
Okay. All right. So can you see my screen?
Okay. Yeah. So
Okay.
next, we are pleased to welcome Professor Kim of KAIST Graduate School of Medical Science and Engineering and CEO of iBeam Technology. In this session titled Seeing in Vivo Immune Responses, the Power of Intravital Microscopy, he will demonstrate how intravital imaging enables real-time observation of immune responses within living systems. Please continue submitting your question in the Q&A box at any time. We will address them during the final Q&A session. Professor Kim, please begin.
All right. So thank you. Thank you for everyone for joining today's webinar. Okay. So as I introduced, I have one GY to declare here. Okay. So Professor Jung already showed some result of the intravital microscopy, right? So intravital microscopy is a technique for microscale cellular level imaging and video acquisition in a live animal model. So this is one, example. So obtained from the acute lung injury model mouse in the lung in vivo. So this is a 10-minute video. So as you can see in this video, we can clearly see the increased activity of the neutrophil in terms of their migrations and the interaction with each others, right? And then as you can see here in the middle, so we can quickly see the clustering of the several neutrophil inside this vessel in the lung can induce microcirculation disturbance, dysfunction, as you can see in here. Okay, so not just this, we can also utilize the real-time intravital microscopy for the delivery of the nanoparticle or any other drugs in a live animal model in vivo. So compared to other imaging modalities such as eye-based system, which can see the delivery of some presently labeled drugs in the liver, as you can see in here. Compared to this, so by using intravital microscopy, we can obtain much higher resolution videos to monitor the real-time drug delivery to certain organ. In this case, it's liver. So as you can see in here, this red color is the intravenously injected present nanoparticle. And as you can see, as this video starts again, we can nicely see how this nanoparticle flows into the liver sinusoid and then distribute and then accumulate inside the liver sinusoid real time in vivo. Okay, so these two examples nicely shows that by using intravital microscopy, we can acquire this live several level image and video live in vivo animal model with some molecular and also functional informations. So for this purpose, IBIM technology, we are providing this dual mode intravital confocal and two-photon microscope. So both of confocal and two-photon microscope is integrated into this compact sized all-in-one packagings. And then everything is optimized for in vivo, live animal intravital imaging. So this microscope can do both of four-color, two-photon imaging and four-color confocal imaging by using this single box. And then we can also, easily connect other, conventional lasers through the optical port behind this box, such as, tunable, titanium sapphire pentasecond burst laser system. And then also this microscope box is equipped with a very high-speed laser scanner, enabling the real-time imaging with a speed of 50 frames per second with 512 by 512 pixels. And then based on this real-time imaging capability, we also built in the automatic live tissue motion compensation function. So this is how it looks like. So this front sliding door can be easily opened, and then you can put the anesthetized mouse model inside under the objective lens. And then everything inside the stage and other things is all heated. So of this whole internal space is heated to maintain the mouse body temperature, animal body temperature to be stable during the long-term intravital imaging. Also inside we have a CCTV camera for real-time animal monitoring to monitor their anesthetization and their status during any time during the intravital imaging anytime. Okay, let me show you just one few example of this four-color, two-photon visualization of a live animal model. So this particular example was obtained from the skin of anesthetized H2B-GFP and MTMG mouse model. So we can nicely see the nuclei of the individual cells in the cell membranes. And also we intravenously inject some other dye to see the blood flow in blue pseudo colors. And at the same time, we can also nicely visualize the dermal collagen by detecting the second harmony generation signal, which is shown in white color here. And also this is another live images obtained from the pancreas of the live anesthetized mouse model. So as Professor Zhang already showed you, pancreas is quite important organ with several fatal diseases such as PDAC. And then again, we can nicely see the individual cell nuclei, their membrane, and most of these are the alginate cell and alginers in the pancreas. And then by obtaining multiple images at different locations in zigzag pattern, we can nicely stitch all those images to make this wide area mosaic images automatically. So this same manner, we can visualize, almost every organ and tissue, live anesthetized mouse model, as you can see in this slide. So we have been able to images, bone marrow, brain tumor, or skin, various tumor model. This particular example is a colorectal tumor model. And then pancreas, spleen, GI tract, adipose tissue, placenta, prostate, lymph node, kidney, muscle, liver, heart, lung, and so on. So to obtain and all these images, I want to emphasize again, all these images were obtained from the live animal model. And then to obtain those high resolution images from the moving tissue in a live mouse model, we definitely need this live tissue motion compensation functions, which is because even though the mouse is anesthetized during the imaging, still it breathes air, and then the heart is beating, and then all those physiological things induce huge motion artifact in the tissue, as you can see in this real-time video. So our approach is quite simple because we can achieve real-time video rate imaging capability. We just record all those frames, all those real-time videos, and then we analyze frame by frame, and then remove the out of focus frames, and then select the in focus frames, and automatically do the motion image registration to compensate the tissue motions. So this is the result. So after this motion compensation, we can nicely average the multiple frames to obtain this high resolution, high SNR images from the moving tissue. And without this motion compensation, you will end up in these blurred images. Of course, in the first place, you first need the real-time intravital microscope to begin with, to begin this motion compensation. Okay, but anyway, so this automatic motion compensation function is built into our microscope software. So all you need to do is just turn on this motion compensation function and just press single button of image capture, and then it automatically captures the real-time video and then do the image registration and then generated this motion compensated, average the frame, the images. Okay. And then if you have more time for post-processing, you can also use this, very new, AI denoising algorithm. So which was already published almost 3 years ago in collaboration with other professor at KAIST, Professor Yoon. But anyway, the good thing about this AI, there are many AI denoising algorithms, of course, but this one is particularly useful because it does not need any pre-training, and then it can be performed unsupervised manner. So which means we can do it automatically. And this is the result. So on the right-hand side, this is the low real-time video obtained from the skin of anesthetized mouse model. And then to see the fast dynamics, we intravenously inject the fluorescent red blood cell shown in blue color. And then after AI denoising, we can improve the signal to noise ratio more than hundreds of times, and then make this very clean, high-speed real-time videos. Another example, and you can nicely see these multiple, flowing red blood cells, and then in these, denoised images. And then this algorithm works very nicely with more noise videos, such as this one. So this one is the real-time video obtained from the kidney. And after the AI denoising, we can nicely appreciate this, subcellular level, tubular structure in the kidney. This is the brain cases. This is a G-camp mouse model. It's very noisy. We intentionally reduce the, we intentionally obtain these very noisy videos, but after denoising, we can very nicely identify the individual cell and dendrites, and from this very noisy real-time videos. And then this software can be also do automatic denoising. So we make this software which we call AI denoisers, iBeam AI denoiser. And then if you are interested, please ask us for a trial. And then you can deal with any video format. Okay, let me go back. I'd like to show the several examples of how we visualize all these different organs. So in the first slide, I showed you the real-time visualization of the lung. And for that purpose, we make this stabilized lung imaging chamber. So in here, by sucking out the air from the chamber, we can build up the very gentle negative air pressure inside it. And by doing that, we can stabilize the this lung surface to the glass. And after this stabilization, in addition, we apply the motion compensation, and then we can obtain this high resolution timeless video from the live mouse model. And then we can directly see how these neutrophils are interact with each other and then make a cluster and disturb this blood circulation in here. So this is the procedure, just for fun to show you. So first we need to intubate our mouse model like this, and then we connect the mouse to the mechanical ventilator, and then we do the small surgery to expose the lung, and then stabilize the lung surface, as you can see in here. And then we put it under our heated stages and the heated space into our microscope, and they put it in, and then this whole stage can be manipulated by using this motorized stages. And then let me show you some immune cell trafficking imaging result as well. So this is the visualization of the trafficking of the neutrophil and macrophages in the ear skin. And in this particular example, we use this LyGM GFP mouse model, so we don't need to label these immune cells. But in red color, this is the vessel. So to label the vessel, we inject the antibody, anti-CD31 antibody conjugate with the Farad flow 4 intravenously at 30 minutes before the imagings. And then it nicely labeled all the endocellular cell, and then allowing us to visualize the vessel in this mouse model. And this is another example. We injected some fluorescently labeled protein into the dummies, and then you can see this infiltration of neutrophil and macrophage to the this injection site. And also, we are also interested in the lymph node imaging, of course. So this is how we prepared our mouse model. So we exported this popped lymph node, and then maintain the tissue temperature to be stable by using additional heating systems. And then this is a 2-hour video, showing the extravasation of a T cell and B cell at the high resolution venue inside the lymph node. And then we can nicely track the extravasation process of this T cell and B cell from the HB lumen to the lymph node parenchyma, as you can see in this video. So this is another video. So this red color is endocellular cell of HB, so which is very plumpy and very thick. And then if you look at this green T cell, we can see how they attach to the endocellular cell and then penetrate between the in the paracellular spaces, and then get into the this the base between the endocellular cell and the basement membrane, and then eventually do the extravasation. If you look at here, you can see this extravasating T cells. So the immune cell trafficking can be very nicely visualized by using our intravital microscope and 2-photon and confocal microscope. Also, it can be quite useful, so as I showed in the second slide. Also, we can nicely use intravital microscope to visualize the drug delivery as well. So this is the one example. So we inject some therapeutic oxygen through the tail vein, and then we can monitor their delivery in the liver to these LyGM positive cells. So this is 10 minutes after, 30 minutes after, and this red color is the intravenous injected oxygen, and then we can see the attachment to the surface and their internalization at 30 minutes. Okay, and then I'd like to show you, several, imaging window technique for longitudinal, repeated, in vivo imaging of abdominal organs. So basically, it's already published work by other groups. So basically, it's ring-shaped windows, and then we designed an additional, holding system. So to make a long story short, so basically it's a ring-shaped metal, and then it can be permanently implanted on the abdominal imaging window, as you can see in here. And after we implant it like this in the to see the kidney, we can very nicely see the kidney tubular structure in cellular level, and then we can repeatedly find out the same tubule from day 0 to 1, 3, 5 to 7, and then we can see we can nicely track the single tubule in the kidney. Okay, and then of course you can imagine after establishing this abdominal imaging window, we can also do many things. We can induce disease. We can also monitor the drug delivery, whatever. And then also, we make another things for PDAC monitoring. So in here, we put the spleen and pancreas in the middle of our imaging window, as you can see here. And then at the time of implanting this pancreatic imaging window, we can inject a mouse pancreatic cancer cell to make orthotopic, PDAC model. And then we do the, repeated imaging and then follow the growth of these tumors. And at the same time, as I explained to you before, by injecting some antibody, CD31 antibody, we can monitor the vessel. So this is the, daily images from day 11 to day 18. And then we can nicely see the growth of this pancreatic tumor cell, and then the changes in the tumor vessel, as you can see in here. Finally, I'd like to show you some example of brain longitudinal imaging technique, which we call cranial imaging window. So again, this is a very well established protocol. So basically, we make a small hole in the skull using drill, and then we put the transparent glass on top of it on the exposed brain. So let me like this. So make a hole, put the glass, and then protect all the exposed area with the dental cement. After this surgery, the mouse can survive for more than 1 year. So we can do very long-term, repeated longitudinal imagings. And after this surgery, again, we can put our mouse into our microscope and then obtain this kind of 3D visualizations. And then during this process, during this procedure, we can save the dura. And then by doing that, we can see the dura in the subarachnoid space, the leptomeninges, pia mater, and then also cortex. This is another example. Again, this is live animal imagings through the cranial imaging window. And then in this particular example, we use NG2DS LED mouse. So we can nicely see the smooth muscle cell and the pericyte in the capillaries. And then from the dura to all the way into the cortex. And then again, after this surgery of cranial imaging window, we can, image the mouse model as much as we want, as long as we want. So this is one example. So in this particular example, we test some BBB shuttle. So on the upper panel, this is a non-control drugs, control antibody drugs. So it just stays in the vessel and never get out. But after applying this BBB shuttle, we can see their delivery into the brain across the blood-brain barrier, as you can see in here. So again, by using the cranial imaging window, we can find the exact same vessel in the brain, and then by imaging this CXACR1 GFP mouse model, we can find the microglia, the same microglia at the same brain area at different time point, like day 0 to day 1. And then we can see their activation, and then we can also monitor, their activity like this. So we are not just providing the intravital microscope, intravital 2-photon compact microscope. We also provide all these, imaging solutions and kit optimized with our microscope. Okay, so I only explained about this one, and then we recently made other version, which we call FS model. So it has a much larger open space to accommodate the larger animals such as rat or guinea pig when parrot. And then even in this the large animal model, still we can nicely achieve this motion compensated cellular resolution images. And then also software is important. right? So we also provide intravital image processing and visualization software called iBeam Studio. And then by using it, we can do automatic, image stitching to make these large area images like this one. And then we can also deal with the human organoid sample in 3D. So this is one example. So this is a 3D rendered image, although this is not in vivo data. This is ex vivo data, but this software can nicely deal with the 3D data. And also, it can deal with the 4D data actually. So 3D images obtained at different time point. And then we can make these 4D videos to observe the movement of the cell, immune cells. This one is just on cells in the skin, and then we can see how they move around in 3D tissue space in a live animal model. So to summarize, intravital microscope quite useful technique for 3D cellular level imaging in the live animal model. So we can do this dynamic imaging of cells, of cellular dynamics, such as cell trafficking, cell to cell, cell to microenvironment interactions, all in the live animal model at many different organs. And also for the drug development, we can use intravital microscope for the in vivo drug imaging analysis. So we can do drug delivery monitoring to the target cell, drug efficacy monitoring, and then validation of the MOA in a live animal model. And then also we provide the imaging service, intravital imaging service. So if you have some experiment you want to do, you can ask us to do that. So if you're interested in this intravital microscopy, intravital two-photon confocal microscopy technique, and all these imaging solutions, please visit our webpage, www.ibimtech.com, or simply send us an email, the information at ibimtech.com. Thank you very much for your attention.
Thank you, Professor Kim. So there's several questions. So first of all, have you also done any studies to ask if LMPs can get into tumors readily? Yeah.
Okay, so yeah, sure. I'm sorry. Yeah, we haven't done it. Let me show you one example. So this is one example. So in this case, we injected some nanoparticle, and then we can nicely see. So this is the longitudinal image at 2 hours, 6 hours, 24 hours of imaging. And then we can see this red dot nanoparticle indeed delivered to the tumor cell in this mouse model. So this is one example. So we can also image the LNP, exosome, other nanoparticles, or even other antibody drugs, and then to monitor their delivery to the single tumor cell level in a live animal model. Okay, so let me move on to the next questions. So does your mouse need any surgery for lung or liver imaging? Yes, I think I showed you some examples before. So we need some surgery to expose our mouse model to see the lung or liver. Yeah, like this one. Mm-hmm. So for kidney, it can be in a similar manner. So we can, to see the liver, we also need to do this kind of surgery. Not just the imaging model, imaging window, we can also do simple incision model, incision surgery to expose our target organ. Okay, and then there are the next question is, do we need a surgery to image the specific organs? Yeah, again, the answer is yes. So we need the penetration of our two-photon and confocal microscope is about 200 micrometer to 500 micrometer maximum. So which means we cannot penetrate the skin. So thereby we just need to expose our target organ by the skin incision. Okay, and then the next question is which mouse strain have been used to capture brain tumor and other in-depth tumor? Can we see same images with the C57 bulb, black C57 bulbous mouse? Oh yes, of course. We have used the black cis mouse model, bulbous mouse model, C57. Yeah, all these strange mouse have been imaged by using our microscope. And then, yeah, of course, for mouse tumor, syngeneic mouse tumor model, we try to match their background, such as B6 and bulb C, depending on the tumor cell line we use. And the next question is how deep the light can penetrate the tissue. Well, yeah, I explained. Our microscope can penetrate from 200 micrometer to 500 micrometer. The penetration depth is different from every organs. So yeah, like this. So for brain, we can penetrate up to 500 micrometer. And then some other organs such as skin, we normally penetrate in 100 to 200 micrometer, depending on the locations. Okay, do you have a list of CROs that utilize your system? Yes, we do. We have one USA-based CRO that utilize our imaging system located in East Coast. So I can let you know, after this webinar. And then they are providing the CRO service using our microscope in the state. And the next question is, how about imaging bone? Yes. So in here, if you look at here, this is the bone marrow imaging. So imaging the bone, well, that's not difficult, at least for the cranium. So for the cranial imaging, we just need the incision on the head, then we can nicely see the cranial bone. And then by using two-photon microscope, we can easily detect the bone structure by detecting the second emergence signal. And then we can penetrate the bone about 100 micrometer, 100 or 120 micrometer, which is enough to see the bone marrow in a thin bone, such as cranial bone. And if you want to image the femur or tibia, then we need a little bit more invasive surgery to expose the tibia or femur. But still, after exposing it, we can do bone thinning to see the bone marrow if you want to see the bone marrow in that. Okay, and then next question is how to keep the mice alive during long time imaging. Well, for like several hours imaging, we just keep the mouse to be anesthetized. And then for several days imaging, we normally image the mouse, anesthetize the mouse, and then do the imaging. And then we walk up the mouse and then put it into the cage. And then, and the next day we bring the mouse again and then do the imaging again. Yeah, so that's how we do. Yeah. So for example, in here, this is 7-day images. So we don't make the mouse to be anesthetized for 7 days continuously. So basically, every day we anesthetize the mouse and then do the imagings. And then next question is, have you used the system to monitor BBB, leakage in the brain tumor model? Yes, we did. Yeah, although I didn't show it in today's seminars, but we have a maker, orthotopic, glioma model by using cell lines, and then we can image the blood-brain barrier and the vessel. So yes, it's possible. We have done it. And then, okay, the next question is how long one imaging session take to capture one images? Well, it depends on the which organ you want to image. So normally surgery takes like 20 minutes most. Yeah, 10 to 20 minutes to properly expose the organ we want to image. And then boom, we can take the image right after those surgery. So within 20 minutes, we can start and then finish the image acquisition from the internal organs. Okay, and then the next question is, oh yeah, Professor Hung-Gi-sung, Professor Sung, thank you. And then his question is, how long can an experimental animal be maintained for a single imaging session? Okay, so by using, so it also depends, So yeah, so as I showed in here, these days we heavily use this isoprene to mouse to be anesthetized. And in this condition, we can keep the mouse to be anesthetized to more than several days. Although we need to provide the water and nutrition as long as we want. So by using injection anesthesia, we can easily make the mouse to be anesthetized for several hours, like 2 hours to 6 hours. And then if we use isoprene, then we can keep the mouse to be anesthetized, more than that, more than 24 hours. And then our maximum was, 72 hours. Okay, and then the next question is, compared to other normal tissue, is there less ZMPs getting into tumor from that tissue type? Well, this question, I'm not sure. Yeah, I'm not sure. And then for this purpose, I think this IBIS optical system would be, better technique to compare the, the difference in terms of LNP delivery to between different organs. But the bottom line is we can monitor the this nanoparticle level delivery in cellular level. Okay, and then
time's up, I think. So
okay. Sorry.
so thank you for submitting your question a lot at the Q&A box. And if you leave your remaining question by our email information@ibmtech.com, we will respond the question. We will respond the questions. Yeah, following.
Okay. So the last question was, yeah, we have a model for large animal imaging, so such as non-human primate. So we put our imaging head into this robotic arm, then we can move it to anywhere we want to image this, and it can be very useful microscope for large animal imaging, including non-human primate. Okay, so anyway, the time's up. So
yeah,
if you ever more questions, please send us an email at information@ibmtech.com.
Yeah, thank you. Thank you everyone for your thoughtful questions and active participation. Before we conclude, we'd like to share a few upcoming opportunities for you interested in hands-on in vivo imaging. From February 24th to 27th, IBM will host an on-site workshop at UMass Chan Medical School in the United States, featuring an intravital imaging seminar and multiple hands-on demo sessions. This is a great opportunity for you to image organs that aligns with your own research interests and experience real intravital imaging workflows using IBM systems. Registration is available via RSVP. Simply click the Request Demo button in the Resource tab below. And we are planning an additional IBM workshop in Chicago this April. So stay tuned for more details and upcoming announcements. If you'd like to explore more information and application example, please visit the IBM Technology website through the link available in the Resource tab below as well. Thank you again for joining today's webinar. We truly appreciate your time and interest, and we hope to see you at an upcoming IBM event soon. Have a great day. Bye-bye.
Thank you.